We just talked about proteins, how proteins are made from the messenger RNA. They are translated. Not sometimes proteins are not made in the correct way, the way they were supposed to be. And that is due to a mutation in the DNA. Mutations are changes in the genetic material, DNA, which are heritable, which are passed from one generation to the next. There can be two different types of mutations. One, somatic mutations. These occur in any body cell, any cell of the body that is not ultimately going to form gametes. When a somatic cell suffers a mutation, a change in DNA, all of its subsequent daughter cells will also have that mutation. When a mutation occurs in germline cells, in cells that will ultimately form gametes, these type of mutations are heritable. They are transmitted from one generation to the next. We are going to talk about specific type of mutation in detail in this module. Those are the point mutations. These mutations are changes of single nucleotide base. Conversely, there are also changes in heredity material at much bigger scale, in more gross form. And those are the chromosomal mutations which we talked about when we were studying genetics. These could be changes in position or orientation of a segment of DNA or deletion of a segment of DNA. We talked about some of these: translocations, deletions, inversions, etc. So we'll look at a couple of categories of point mutations. The first one is silent mutation. When this type of mutation happens, this type of change in a single nucleotide of DNA, genetic material happens, there is no change in the protein which is ultimately formed from that segment of DNA. And that is due to redundancy of genetic code. We looked at the genetic code and we saw that there were several codons which were specifying a specific amino acid. So here's an example: mutation at position number 12 in the DNA example given here. There is an A instead of C. So a C gets changed somehow into an A. So when there is transcription and ultimately translation, the place where the leucine is supposed to be, it is still the proline. It has not changed the amino acid, although there is a change in DNA, but resulting protein does not have a changed amino acid. So there is no change in the protein function, or this type of mutation will not generally be detectable since it's not altering the protein activity. There are missense mutations. There are base substitution mutations that change the genetic message. For example, when one nucleotide is switched with a different nucleotide, the protein that is formed subsequently as a result of transcription and translation, that protein now has a different amino acid than it was originally intended. In the example here, mutation at position number 14 in DNA, there is an A instead of T. So as a result, RNA is formed, and when RNA is translated, we end up having a valine instead of aspartic acid. So this would change the structure and function of the protein. Even a single nucleotide change can have drastic effects on the physiology of the cell and, of course, the organism which is hosting that particular change. So let's look at an example of this. Sickle cell anemia, in which there is in messenger RNA which is coding for a glutamic acid at a particular position, is changed to valine. The result is this happens in the gene for beta globin, and when this happens, the ability of beta globin protein to respond to low oxygen concentration changes. In normal, beta globin protein will not have any problems if the oxygen concentration is low, which can happen especially in muscle cells when we are around muscle cells when we are exerting. This beta globin protein accumulates, bundles up, forms these bundles under low oxygen conditions, and results in formation of the shape of the cell changes. For example, here, look at this one. This is sickle cell phenotype. It's a red blood cells. This is what happens when the cell has beta globin protein, which is defective because of a mutation of a single nucleotide. And here is a normal red blood cell. So this is an example. Now let's talk about nonsense mutations. These base substitutions result in stop codon. To form, we know that we have seen the genetic code. There are 3 stop codons. So if a codon which was originally specifying a particular amino acid in a protein is changed due to single base change, is changed into a stop codon, it could result in formation of a much smaller protein, and obviously smaller proteins may not be able to carry out the functions of the full normal protein. Here, for example, mutation at position number 5 in DNA, there's a T instead of C. And because of that, we have a codon that, this is the stop codon right here, UAG. And the first amino acid we know is always a methionine in the protein. After that, there's a stop codon. So because of that, this protein will never be made. And that can also have severe effects than the other 2 types of mutations we have talked about. They're frame shift mutations. Codons is a set of 3 nucleotides, sequence of 3 nucleotides. And this is how it is being read by the ribosome, 3 nucleotides at the time. So if there's an addition or deletion of a single nucleotide, the whole reading frame is altered. And because of that, new codons basically appear. They change from the original version. There is a change. And wherever there is an insertion or deletion of a single nucleotide, all the amino acids that will be incorporated in the protein will be different from the ones which were being originally coded. So it can also have a very dramatic effect. The example here, mutation by insertion of T between bases 6 and 7. So there is a T nucleotide inserted between bases 6 and 7. So as we have seen, the nucleotide codons, our reading frame is 3 nucleotides at a time. Here's a 3 nucleotide, here's a 3 nucleotide. So when we add an additional nucleotide, the whole sense of this message is altered. And all the amino acids which will be subsequently added during protein synthesis, now they will be different from the ones which were supposed to be in the wild type or the normal protein. So these are the frame shift mutations. I would like to mention that generally we think of all mutations as being bad. That's not always the case. There are some mutations that can give advantages to the host organism. For example, I told you we talked about sickle cell anemia, in which beta globin gene has a mutation, a point mutation, resulting in a different type of cell, and it behaves abnormally. It so happens that people who have this mutation are also relatively more resistant to malaria. So I'm sure you guessed it, that this gene is more predominant in areas where there was prevalence of malaria. For example, people from Africa have this gene. This gene is more common in people from African nations. Also, these mutations result in diversity in the population, and these mutations are basically the fodder which fueled the evolution. All the diversity we see in living organisms is due to these mutations that gave rise to different organisms, and then the nature selected. We know the Darwin's theory, and that resulted in formation of new species. So mutations can alter the function of a protein. Ultimately, they can have dramatic effect, good or bad, on the organism that is carrying that mutation.